Start with the established triad: tau tangles in the neuron, activated microglia in the tissue, and amyloid plaques forming the dense deposits. This has been the standard pathological map of Alzheimer's disease for decades. In this model, the cell's internal recycling system, autophagy, is treated as a secondary player. It is viewed as a background clearance pathway that simply slows down, allowing amyloid or tau to pile up. The evidence points to a deeper structural breakdown. Autophagy provides the physical substrate on which the entire half-century progression of the disease runs. This diagram maps the strict directional pipeline of macroautophagy. It begins with initiation, where a membrane nucleates around a target. That membrane must then close to form a sealed autophagosome. The sealed compartment fuses with a lysosome, which must then acidify. Only then can the final step, degradation of the cargo, occur. The genetics of Alzheimer's map directly onto this specific pipeline. Take BIN1, the second strongest common genetic risk factor for late-onset disease. The BIN1 risk variant physically impairs the sealing of the autophagosome, halting the pipeline at the closure stage. At the opposite end of the pipeline sits proselenin. While known for its role in early-onset disease, proselenin is required to deliver and assemble the pump that acidifies the lysosome. A mutation here means the VATPase pump fails to drop the pH, and degradation halts. We have two independent genetic lesions acting at opposite ends of a single machine. One cannot close the bag, the other cannot dissolve its contents. Both produce the same biological outcome, the lethal accumulation of undegraded cellular cargo. Alzheimer's presents as a unified spatial and temporal front of clearance failure. The proteins found at autopsy represent the uncollected cargo of a single broken machine. This failure begins in the locus coeruleus, a small nucleus in the brainstem. The neurons here are autonomously pacemaking. They fire tonically throughout waking life, demanding high ATP production and placing extreme structural strain on their mitochondrial networks. Operating at the absolute ceiling of their metabolic capacity means these cells have zero reserve. Their survival relies on retiring damaged mitochondria before toxic byproducts accumulate, a highly specialized branch of clearance called mitophagy. Mitophagy is an ATP-expensive process, and it breaks down here in two ways. First is energetic. As the central redox currency NAD+ declines with age, the sirtuin signaling network starves. This silences T-Feb, the master regulator commanding the transcription of new lysosomes. The cell stops manufacturing the structures it needs to clean itself. The second blow is structural. Amyloid beta physically binds to and throttles TOM40, the central protein import channel on the outer membrane of the mitochondrion. Once obstructed, the organelle is rendered unrepairable. It can no longer import the nuclear-encoded proteins required to maintain itself. This import blockage also breaks the cell's warning system. PINK1 kinase fails to properly interact with the machinery. The Parkin clearance system never recognizes the danger. Phase 1 creates a localized biological disaster. The locus coeruleus is left holding a growing population of toxic, unrecognizable mitochondria. Their unchecked reactive oxygen output provides the inflammatory tinder that catches fire in the next phase of the disease. As pathology moves into the hippocampus, the cellular environment changes, and so does the substrate load. The primary actors are now microglia, the brain's immune cells, tasked with ingesting high volumes of myelin debris and apoptotic membranes. To survive this volume of intake, the microglion must physically process the ingested lipid through lipophagy, relying on lysosomal acid lipases to break down the dense fat droplets. When the volume of debris exceeds the internal clearance capacity, the cell gridlocks. It transitions into the lipid droplet accumulating microglia state, known as LDAM, where it becomes entirely engorged with undigested lipid. Pushed past their physical limits, lysosomal membranes destabilize and rupture, spilling destructive cathepsins and mitochondrial damps into the microgliocytosol. The presence of these specific cytosolic damps acts as a canonical second signal. It arms the NLRP3 inflammasome, shifting the cell aggressively into a hostile effector state. In the third phase, the disease reaches the cortical neuron. Here, the autophagic system physically merges with the endosomal sorting pathway, operating under a heavy proteostatic load. We see this shared machinery collapse in real time through the ESCRT3 protein complex. This same complex fails at two distinct addresses, halting autophagosome closure on one side and failing to sort the endosomal multivesicular body on the other, a unified failure. And here, damage from previous phases catches up. Unchecked lipid peroxidation from phase 1 and 2 generates reactive aldehydes, specifically 4-HNE. These molecules form covalent adducts directly onto the structural subunits of the lysosomal VATPase proton pump. The pump halts. The lumen fails to reach the critical pH threshold of 4.5, and hydrolase activation ceases. Degradation is completely offline. The morphological result is the panthos lesion. The neuron distends into a toxic, flower-like rosette, packed with swollen, A-beta-filled autolysosomes it cannot digest. The swelling continues until the panthos cell finally ruptures, extruding its undigested intracellular amyloid mass directly into the surrounding tissue. The extracellular amyloid plaque is the extruded residue of a neuron that died of autophagic failure. The lethal breakdown happened inside the cell, years before the plaque appeared. This model reframes cognitive resilience entirely. Resilient brains tolerate heavy protein burdens because their autophagic clearance pipelines remain structurally uncompromised. They continually dispose of damaged components before they cross into a fatal loop. A disease of failed clearance requires restoring clearance, but generalized autophagy enhancement is a blunt and frequently dangerous tool. The intervention must match the exact temporal phase of the breakdown. In phase 1 and 2, with downstream structures idling, the goal is feeding the machine. Supplying NAD+ precursors restores energy, unlocking new clearance capacity. In phase 3, iron chelators are required to protect the pump, preventing the 4-HNE adduction that poisons the degradation stage. Mismatching these therapies is disastrous. Applying an mTORC1 inhibitor to induce clearance flux in a phase 3 cell with a poisoned lysosomal pump does not help the cell. Forcing upstream creation of autophagosomes into a blocked downstream pathway accelerates the lethal panthos spelling, tearing the cell apart faster than the disease alone. The cell that cannot clear itself is the absolute center of Alzheimer's disease. Effective treatment relies on matching the molecular intervention to the exact temporal phase of the collapse.